Yes, I am not audible. I think so now I am audible, right? Let me check once. Let me check once. Let me check once, guys. Yes, I am not audible. Okay, great, great. So now I am audible and let's get back to the topic. So the session is starting right now. The session is starting right now. Let me clear all the things again. So in today's session, what we are going to discuss, we were going to discuss about the photosynthesis, right? In today's session, the photosynthesis we are going to discuss. And first of all, let me tell you that how photosynthesis or what is the reason for photosynthesis. So the reason for the photosynthesis is basically what the plants wants to make glucose, okay? This is why the plants are doing photosynthesis. Now, the process of formation of glucose doesn't take place in a single step. It takes place in multiple steps. And those steps are basically what? Those steps are two steps, which is number one, light reaction, and the second one is the dark reaction. Clear? So these are the two steps in which glucose formation is taking place through photosynthesis. Clear? In light reaction, guys, what is happening? In light reaction, sunlight is captured, and with the help of that sunlight, first of all, the plant is making ATP. And you know, ATP is what? ATP is the energy currency of the cell. Now, in dark reaction, the plant is going to form glucose. So this is clear. In today's class, we will be focusing on the light reaction, and if we have time, then we will be going to move forward. Otherwise, we will finish the session on light reaction only. Then dark reaction we will do in the next session. Great. This much I have discussed when I was on mute. So sorry for the inconvenience. Now, let's have the chloroplast. So first of all, let us understand that where does photosynthesis takes place, where does light reaction takes place, and where does all these pigments and all are present. Okay, we discussed the structure of the chloroplast day before yesterday. So this is the membrane of chloroplast. Okay, this is the membrane of chloroplast. And you remember that this structure we were calling it as grana. Yes or no? This structure we were calling it as grana. And the single ones were known as what? The single ones were known as thylakoid. The single ones were known as thylakoid. I hope that this thing is clear to everyone. Thylakoid. Ignore my spellings, okay? Thylakoid. Clear? Okay, so all these pigments like chlorophyll A, chlorophyll B, xanthophylls, carotenoids, everything, every pigments, where it is present? It is present in the thylakoids. They are present where, sir? They are present in the thylakoid. Okay. In case you have any kinds of doubt, let me know in the comment section, because today we are going to clear this Z scheme, and I promise that you will not have any kinds of doubt after this session. So all the pigments are present in the thylakoid. Okay. Now, after moving forward, let us see the thylakoid. This structure is just the thylakoid. So I can see that this is the membrane of thylakoid. Okay. Outside the thylakoid, what is present? See here. Outside the thylakoid, we have the stroma. Outside the thylakoid, we have the stroma of chloroplast. Okay. We have the stroma. This much is clear. And what is this structure, sir? Inside the thylakoid, we call there is some space inside the thylakoid. See, if you can't see, I'll zoom it for you. See, this is what? This is a structure. This is a space. So this space will be known as what? This space will be known as lumen. Remember that a small step at a time will make our concept very clear. Don't just jump to the main concept directly. We will do small step, and you won't be, you will be like, oh, that's what the Z scheme was. Yes. So this was the lumen, and the outer portion is stroma. Now just remember, if I talk about some cell membrane, so what things comes into your mind? If I ask you about the structure of the cell membrane, so you will be like, sir, the structure of cell membrane is made up of kind of lipid and something like that, right? and we have read in class 11th about the cell chapter. In the cell chapter, we have read the structure. So we are going to draw that structure. That means wherever I am talking about the cell membrane, all cell membrane will be same. Yes, so I am drawing the cell membrane of the thylakoid. I am drawing the cell membrane of the thylakoid. Clear? Okay. So you know the cell membrane is made up of lipid bilayer, right? So we have this. Yes or no? These are the molecules of lipid, and they're arranged somewhat like this. They are arranged somewhat like this. Okay, great, sir. Now what? Now I am going to show you where exactly the pigments are present. Now the pigments are present in these bilayers. The pigment are present in these bilayers. See, let's make one pigment here. Oh, what happened? iPad, draw a square. Sorry, this is a rectangle. So one pigment, let's suppose this is there, okay? Now let's have the another pigment. Let's suppose this is the another pigment. So can you see how they are arranged? How these pigments are arranged? So basically they are arranged in the membranes of the thylakoid. Now you might have a clear visualization like where the pigments are. All the pigments, chlorophyll A, chlorophyll B, xanthophylls, carotenoids, all the pigments, where they are present, sir? They are present in this manner. What is this? This is the membrane of the thylakoid. This is the membrane of the thylakoid. Now, I am going to change this image in a simple manner. I am going to change this image in a very simple manner. Can I draw a line here? Can I draw another line? And remember, these two lines are representing what? These two lines are representing the lipid bimolecule layer, okay? These two lines are representing what? These two lines are representing lipid bimolecular layer, okay? So I'll just remove this lipid structure so we won't complex our diagram, okay? Now, here I am making these pigments, okay? With another color I'll use, let's take red, okay? Here are the pigments we have. So one pigment here, one pigment here. Clear? This is how the pigments are arranged. Here what it is? This is what? This membrane is the thylakoid membrane. Yes, this membrane is the membrane of the thylakoid. Outside this membrane, what is present, sir? Outside the membrane, stroma is present. Yes or no? Outside the membrane, stroma is present. Clear? And inside this side, the down part, what is present in the down part? In the down part, sir, we have the lumen of thylakoid. Yes or no? We have the lumen of thylakoid. Clear? So what I have done is that I have just made this structure. I have just made this structure. Here was stroma and here is lumen. Clear to everyone? See it clearly. Here was stroma, this was stroma, and this was lumen. So instead of making a single line, I have what? I have made it in the form of lipid bilayer, right? In the form of lipid bilayer, in the form of lipid bilayer. Clear to everyone? Any doubts? Anyone? Let me know in the comment section. If you like this video, if you like the concept, share these videos among with the friends. Clear? Great. Now, let's move on and let us again have some great concepts. Okay? Clear. So, after this what? Each and every pigments. See, each and every pigments, they are basically what? They are basically made up of lots and lots of things. They are having lots and lots of pigments, lots and lots of chlorophyll, xanthophylls, each and everything. Okay, so they are arranged something like this. Now we are dealing with what? We are seeing with these pigments. We are taking these pigments and we are seeing these pigments. Okay, so what happens is that these pigments are made up of lots and lots of these complexes are made up of lots and lots of pigments. Okay, great, sir. Now see, this main one is known as a reaction center. This main one is known as a reaction center. Clear? And these pigments are the other pigments. Maybe chlorophyll B, maybe xanthophylls, maybe some other things, right? This is the main one. This is queen. This is, let's suppose, a queen. This is a main pigment. and all the other pigments are the secondary pigments. Clear? Now what happens that the work of these pigment is what? The work of these pigment is to absorb the sunlight. The work is to absorb the sunlight. Now what they do is that as soon as these pigments absorb the sunlight, what happens? They emit electron. Easy? It's very easy. In chemistry, you might have learned that if you're giving an energy to a molecule, its electron gets excited, right? If it is in n is equal to 1, so it will go into n is equal to, it will go into n equals to 3, right? So similarly here also what's happening, as soon as the pigments are getting the sunlight, their electrons are energized and they are going into its excited state. Clear? This much clear? Okay. Till now what we discussed? We discussed that these are what? These are some of the complex of the pigments, and they are made up of what? They are made up of lots and lots of pigments. In these things, there is one main pigment and the others are what? The others are the pigments which are helping in something, or they are known as the accessory pigments. Okay, right now we just know this. Now we know that if the pigments are receiving the light, the electrons in their pigments will get excited. The electrons in their pigments will get excited. Clear to everyone? Okay. Yes or no? That's great. Any doubts? Let me know in the comment section. Any doubts? Let me know in the comment section. Okay. At least you can say me hi, hello. Yes. If you're watching this video, say hi and hello. Clear. Okay. Now, sir, what? Now, these were known as what? These were known as light harvesting complex. These were known as what? Light harvesting complex. Should I write the full form? Should I write the full form? Okay, let me write it. These were known as light harvesting complex. Okay. These are known as light harvesting complex. Great, sir. This much is clear. Now let's move on. Again, I am talking about the light harvesting complex. Now, in this light harvesting complex, what is going to happen? See, the main pigment is what? The main pigment is chlorophyll A. Okay, the main pigment is chlorophyll A. Now, this chlorophyll is what? This is the main pigment. So, like, you know, if someone says that you are the head of the house, now you have to do your orders will be our superiority kind of thing. So, you will be like, okay, I'll do this. Very easy work you will take, and then you will order someone, bring me a glass of water, you do this, you do that. So this chlorophyll A is also kind of that person. Okay, it is considered to be a main pigment, so it has its own tantrums. It says that I will only and only absorb the light of blue and red color. Okay, clear? Chlorophyll said that I will be absorbing the light of blue and red color only. Then what about the others, sir? What about the others light, like yellow light, orange light, and or whatever the things which is present in the VIBGYOR? What about that? Remember that already we have so much less amount of light, right? We are just photosynthetically active radiation was what? It was 400 to 700, right? And in that also, this particular chlorophyll A is saying I am going to only and only absorb B and R. That means blue and red light. So what is going to happen here? These people are gonna work. Okay, these chote mote pigments are going to work. Carotenoids, xanthophylls, all these are gonna work. They will be like, okay, chill bro, I am going to absorb the light and will send you the electrons. I am going to absorb the light and send you the electrons. So what is going to happen? See here. Let's suppose we'll draw a sun here. Okay, the light is coming, and this particular pigment's electron get, where should I stand? Yeah, I should stand here. This particular pigment's electron gets excited. Now, this electron went here. This electron went to this particular thing. So, all electrons are basically what? They are kind of coming to this particular reaction center. These people don't have any other work. They can't do the actual photosynthesis. They don't have that power. The actual thing is going to be done by the reaction center. But they are kind of helping the reaction center to grab the electrons. Clear? This much is clear to everyone? Yes or no? Is it clear? Great. If this is clear, let's move on. Okay. Now, moving towards the types of photosystem. Okay. Hi, how are you? Shri Sai. Should I pronounce your Shri Sai? So, should I pronounce you Shri Sai? Yes? Great. We will definitely going to crack AIMS. Yes or no? It's my promise that I'm going to make your concepts very clear and we are going to crack AIMS this year, right? Okay. Now, let's have this types of photosystem. Okay, what is this types of photosystem, sir? See, I drew the two diagrams here, but I didn't told anything about the photosystem 1 or 2. Let's discuss that thing now. So, basically, in the membrane of the thylakoid, we have two types of photosystem. Okay, we have two types of photosystem. One is the photosystem 2 and the other one is the photosystem 1. Everything is same. Whatever we discussed here, na, everything is same. This photosystem 2 is made up of same thing, like one reaction center, and these are white are the accessory pigments like xanthophylls and all those things. Here also, photosystem 1 is also made up of the same thing. Reaction center, this. Okay, great. But what is the difference then? Why we are calling it as photosystem 1 and photosystem 2? The major difference is what? The major difference is here. In photosystem 2, the chlorophyll A molecule, which is sitting in the reaction center, the chlorophyll A molecule, which is the main molecule and which is sitting in the reaction center, is what? Is basically of P680. What is this P680, sir? It's a phone number? No, it's not a phone number. This said that I am going to absorb the light of P680 nanometer. Okay. In a similar manner, in photosystem 1, it is having P701. Okay, so this is the major difference. If you're not able to see, I can adjust a little bit too. Okay, this is P700 and this is P60. Major difference between photosystem 1 and photosystem 2 is what? Is photosystem 2 is having reaction center of P680 and photosystem 1 is having the reaction center of P700. Clear? Great, sir. Now, which one was discovered first? Which one was discovered later? So see, first of all, we are going to talk about the photosystem 2, but photosystem 1 was discovered later. That means what? That means if you are going to see, suppose that this is the thylakoid membrane. Okay, this is the thylakoid membrane and you're going to walk first. So first you are going to find photosystem 2. The photosystem 2 will be working first. Okay, then photosystem 1 will be working. But which one was discovered first? The photosystem 1 was discovered first. Okay, understood? Any doubts? Any doubts, Sri Sai? Any doubts? Let me know in the comment section. Okay, what I said, the photosystem 1 was discovered first. But in working, if you see who is working first, so photosystem 2 is working first and then photosystem 1 is working. Okay, so the naming of photosystem 1 and 2 is basically on their discovery, not on their working. So remember, in this question can put up in NEET that what is the basis of naming of photosystem 1 and photosystem 2? On the basis of working or on the basis of their discovery? So you are good children, you are studying with me, so you will mark the on the basis of their working. No, on the basis of their discoveries. Clear? Great. Now why I am looking so pinkish in my video? Okay, your side is now pink. Let's get back here. Okay, so this was the structure of, again let's have a quick recap here. So this was what? This was the chloroplast, the membrane of chloroplast. This was the thylakoid, yes or no? And inside the thylakoid we have the lumen, and this was the stroma. Clear? Now whatever the thing we are going to discuss in this part, this membrane part. Okay, all the things are present here. Photosystem 1, photosystem 2, everything we are going to discuss here. Okay, so again I am going to zoom it out. Again I am going to zoom lots and lots of things. Okay, see. So what I have done is that instead of making this thing, what I have done, I have made two lines. I have removed the lipid bilayer. I have removed your lipid bilayer and I have just made two lines here. okay, just to represent photosystem 2 and photosystem 1. Clear? Great. Now, let's move on and let us talk about the main thing. Now, whatever you are doing right now, whatever you are doing, sorry, what happened? Yep. Yeah. Whatever you are doing right now, forget about everything and now focus on the concept, because this is the thing, this is the main concept which we are going to have. This is the main thing which is going to make your concept strong. Clear? Great, sir. Okay. Now I am making what? I am making the membrane of thylakoid. Okay. So let me have the membrane of thylakoid here. This is the one lipid bimolecular layer, and this is the another lipid bimolecular layer. Remember that I'm not drawing two membranes, right? I'm not drawing outer and inner membrane. I'm just drawing what? I am drawing the one layer, which is this one of lipid bilayer, and the another layer of lipid bilayer. Okay. Don't get confused that thylakoid is double membrane structure. No, never. Thylakoid is what? Great, great. So this is what? This is not a like what do I say? This is not a two double membrane structure. This is a single membrane structure. Just I am trying to represent the bilayer in this form. Okay. Now quickly, let's get started. Quickly, let's get started. Okay. Anyone watching the recording, this is the crucial time. Do whatever you are doing, just leave the things and focus here. So what is happening here is that, first of all, we have this photosystem 2. Okay. First of all, we have this photosystem 2. I'll use little thin pen so that the writings could be easily visible. Photosystem 2. Now what, sir? Now this photosystem 2 received sunlight. Now the photosystem 2 received sunlight. And if it is going to receive sunlight, what is going to happen? Ejection of electron. If it is going to receive the sunlight, ejection of electron is gonna take place. So let's have this particular light came. Okay. And here we have the reaction center. Okay, let's suppose here we have the reaction center, which is chlorophyll A, and they ejected the electron. They ejected the electron. Clear? Nice, sir. Great. The electron is ejected. This photosystem is what? Photosystem 2. If this is photosystem 2, if this is photosystem 2, 1 second, is my screen visible? Yeah, it's totally fine. Yes, it's totally fine. Just a second. There is a little lag in my screen, but it will be fine within few seconds. Okay, don't worry. Don't worry. Is it fine? I think so. Yes, it is. Clear. It will be fine. Don't worry. Yes. Okay, great. So we have what? We have the photosystem 2, and photosystem 2 received the light. Now, this light did what? This light helped in the ejection of electron. This light gave the energy to the PS2, and in PS2 the electrons got ejected. Clear to everyone? Great, sir. Now what? Now, just give me a second. There is my screen lag, so I'll fix it very quickly. There is a little screen lag in my, just a second. I'll check if everything is fine or not. Just a second, people. I'll fix it, okay? Don't worry. Don't worry, this will be fixed. Might be a very little internet connection, I think so. One second, what happened? The class was running so smooth. Suddenly, what happened to our class? Now I think so it's fine, right? Yeah, it's fine. It's pretty much fine now. I am so sorry. Okay, everyone. So let me know if I am live right now again. Let me know that if I'm live. Yes, I am live again. I am live again. I don't know what's happening to this class, but something is wrong with this class. But I now know that I am live again. Okay, clear, right? Chalo, great. If you are facing any kind of delay, it will be fine. Yes, chalo. Let's get back. Okay. So what we were discussing the main thing, and in the main thing only, YouTube was like, oh, you're going to teach the main thing. Let me show some tantrums. Okay. Now we are going to discuss this thing again from the very starting. Okay. So let's have what I just told you about that. I just told you about something which was this thing, right? This particular thing, which was what? Which was the lipid bilayer, right? This was the lipid bilayer, which was photosystem 2 and photosystem 1 we have made. Okay. Now this lipid bilayer, what I did is that I just changed this thing into a curve structure. Okay. I just changed this thing into a curved structure, and here I arranged this photosystem 2. Now I am saying that this photosystem 2 is what? This photosystem 2 is receiving the light. This photosystem 2 is receiving the light. Okay. As soon as this photosystem received light, what is going to happen? The electron is going to be excited. The electron is going to get excited. Clear to everyone? Yes or no? Great. The electron is going to be excited. After this, where will this electron go? Where will this electron go? This electron was calling their molecule that, see, I got energy, now I'm gonna fly high. Now this electron came high and he is not having any kind of idea where to go. So this electron is going to do what? This electron, am I audible properly? I'm having a doubt. Yes. So this electron will be captured by primary electron acceptor. This electron will be captured by the primary electrons acceptor. A kidnapper will come and capture this electron. Okay. This is what? This is the primary electron acceptor. What is the primary electron acceptor, sir? See, primary electron acceptor. The name for this primary electron acceptor is The name for this primary electron acceptor is what? It is pheophytene. What is the name? The name for the primary electron acceptor is pheophytene. Clear? Okay. So the primary electron acceptor, which is pheophytene, is going to capture this particular thing, which is the electron. I'm so sorry guys if you are facing some issues with the session, but let's little bit manage today. Okay, because I really don't want to cancel this class because we are dealing with such an important topic here, but I don't know what's happening. I think so I'm clearly audible. Okay, my voice is clearly audible, my screen is clearly audible, so we can go, right? Let me know guys in the comment section, should I carry on or should I stop the class? Is it fine for everyone? Yes, now it's fine, I believe. Sorry, na? Sorry. So sorry for these kind of delays. Okay, great. Clear. So the electron is going to go where? The electron is going from the photosystem to the primary electron acceptor. Okay, from the photosystem to primary electron acceptor. And this primary electron acceptor, the name for this primary electron acceptor is what? Pheophytene. The name for this primary electron acceptor is pheophytene. Clear to everyone? Yes or no? Great. Great, sir. Now, from the pheophytene, the electron is going to be captured by another thing. The electron is going to be captured by another thing, which is plastoquinone. From the pheophytene, the electron will go to plastoquinone, which is PQ. The electron is going to go towards the plastoquinone. Okay. From the plastoquinone, the electron is going to go towards the cytochrome B6F. Okay, it is going to go cytochrome B6F. Clear? Okay, great. Yes, everyone. So I think so that there will be no problem now. Okay, let's continue the class. So sorry for the disturbance for everyone. Let us get back to the concept. Like I said, that this is the importance concept and everything is happening, right? Okay, now I think so everything is fine. Yes, everything is fine, people. Everything is fine. Chill. Let's carry on with our class. Okay, so the thing which I was saying, the thing which I was explaining was basically what? Was basically the Z scheme. Okay, so what I said that the PS2 will be there to receive the light, and the light is going to help in the ejection of the electron. Like the electron will get excited. Now the electron is excited. Now what will going to happen with that electron? That electron will be captured by the electron capturer or primary electron acceptor. Okay, this electron is going to be captured by a primary electron acceptor, and the name for this primary electron acceptor is what? It is pheophytene. The name for this primary electron acceptor is pheophytene. Okay, done, sir. This much we have understood. Clear? This much we have understood. Now after that, what is going to happen? After that, pheophytene will give the electron to the plastoquinone. After that, this pheophytene is going to give the electron to the plastoquinone. This plastoquinone will be like, I also don't need this electron. I'm also gonna transfer this electron to the cytochrome B6. Okay, cytochrome B6. Okay, so what's happening right now? Learn this thing right away. Learn this thing. Chlorophyll A, PS2, reaction center, light came, electron got excited, electron went to pheophycine. Pheophycine is what? The primary electron acceptor. Primary electron acceptor gave the electron to PQ. PQ gave the electron to the cytochrome B6 complex. And then cytochrome B6 complex gave the electron to PC. What happened right now? Oh my God. Like, now everything is like something is happening to my class. Guys, just a second, okay? I really don't know what happened right now. If not, okay, if not, I will, like for you guys, I will relaunch this class maybe tomorrow. If one second, I'll try to figure it out what's happening with me. That someone came to my class half-hearted, like, oh, okay, sir, we have to attend the biology class, not with full mind, right? That's why it's happening. One second, people. I really don't know what's happening to my class. Now this green screen is not working. Okay, so very sadly, I have to say this that I will going to cancel this class because now I don't think so I will be able to continue this session. So in the next session, what I am going to do is that I am going to start the next session from this topic only, which is the Z scheme, and I'll try to make you understand all the things. I thought that we will be completing this Z scheme today, but now something is happening to my class. Let me check once again if this can be resolved. Okay. I can understand you guys. The topic was very interesting. Everything was going fine, but the YouTube and everything is like, how the class can go smooth. 1 second, okay? I'll try my best to resolve it right now. 1 second. I'll ask my team also. It's done. It's done, guys. It's done. It's done. Now I hope that there will be no issues. From the past 5 to 10 minutes, we're just moving in the Z scheme only. If anything happens now, and then I'll cancel the class. Okay. Hi, Shri Shai. Lots and lots of things happened today in the class, right? Now Shri Shai is also live, so I hope so that there will be no delay. Right? Great, great. Thank you for being with me in the class. Chalo, let's again start. Okay, let us start again. PS2, okay, sunlight. From again, I'm starting with PS2. PS2, sunlight, it received the sunlight, okay. PS2 received the sunlight, and after receiving the sunlight, it ejected the electron. And went where? It went to the primary electron acceptor, which was pheophytene. After the pheophytene, it went to the plastoquinone, and after the plastoquinone, it went to the cytochrome B6, and then it went to where? It went to the plastocyanine. After this cytochrome B6, it went to the plastocyanine. One second, okay. I am resharing my screen. I hope that this time nothing happens. Okay, want to talk seriously with you all, everyone, whoever is present. So let's not waste our time, okay? Your time is very precious, and constantly some things are going on with this, some technical issues. I am facing a lot of technical issues. So I will cancel this class here only, and we will start tomorrow with the, again, which is it fine? No, there is some delay, okay. There is lots of issues going on. So what I will do is that I will schedule your class again at 6:30 tomorrow, okay. I'll schedule your class again at 6:30 tomorrow, and we are going to meet at 6:30, and we are going to complete that Z scheme, okay. I promise you that you will feel like this was so easy. That's my promise. I was supposed to teach that topic today, but unfortunately I tried lots and lots of things, but still we are getting lots and lots of lags and delays. So there might be some issue with my system or the internet. I'll figure it out. I'll reschedule your class at 6:30. Will that be fine? Will that be fine to everyone? Great. Thanks a lot, guys. Thanks a lot for attending my class. And in case you have any doubts or anything, let me know in the comment section. You will get DPPs, you will get your notes, everything in the chat box in the comment section. I will be putting a link of Excel sheet. There you will have my class links, and you will also have the access to your class notes and dpps. It will take some time to make, okay, but it will be available very soon. Okay everyone, till then, bye bye, see you. That's very great, Shri Sai. Let's meet tomorrow at 6:30.